This video is about immunoglobulin, antibody glycoprotein molecules that are produced by plasma cells and can combine with the corresponding AG specifically are called antibody. Antibody is produced by B cells in the response to a stimulation of antigen. Antibody possesses a high degree of specificity and affinity. Immunoglobulin. It refers to all globulins that possess the activity of antibody or show a similar structure to antibody. Therefore, all antibodies are immunoglobulins, but not all immunoglobulins possess the functions of antibodies. And the concepts: why globulin, antiserum, humoral immunity, SLG, secreted and membrane bound, BCR. Okay, sectional molecular structure of Ig, he could see that. Then basic structure, 4 polypeptide chains. Ig is composed of 4 polypeptide chains joined by SS bonds, interchain disulfide bond. Intrachain disulfide bond shows T or Y shape. H and L chain. Heavy chain, light chain. Two terminal ends for each peptide chain, N terminal end and C terminal end. L chains attached to H chains from N end. Classes and types of Ig according to the difference of H chains, amino acid composition sequence. Igs can be divided into 5 classes, subclasses. 5 classes of H: gamma, mu, delta, epsilon. Gamma, alpha, mu, epsilon, delta. 5 classes of Igs. 5 classes of H chain: alpha, mu, gamma, delta, epsilon. 5 classes of Igs: IgG, IgA, IgM, IgD, IgE. IgG1 to IgG4, IgA1, IgA2, IgG, IgD, IgE, IgA, it's a dimer, then IgM, pentamer. Okay, J chain is also there. According to the differences of L chain, 2 types of L chain, kappa and lambda subtypes. L chain difference, kappa, lambda, chain. 2 regions of each peptide chain, constant region and variable region. Variable region, constant region, and hinge region in between. Constant region C, variable region V. Variable region, hypervariable region. There are 3 highly diverse sites within the V region. They are called HVR. Framework region FR. Framework region will be there. Antigen binding sites. Complementarity determining regions CD. Antibody specificity. An individual animal can make billions of different antibody molecules, each with a distinct antigen binding site. Each antibody recognizes its antigen with great specificity. Complementarity determining regions: CDR1, CDR2, CDR3, CDR1, CDR2, CDR3 in L and H. Idiotype of Ig. Igs produced by different B cells possess unique structures respectively in hypervariable region HVR. Igs produced by different B cells possesses unique structure respectively in hypervariable region HVR. The unique structure of Ig is called idiotype or idiotypic determinant. In fact, HVR, CDR, idiotype are in the same sites of Ig. Hinge region, flexible and suitable for CDR of Ig binding to antigenic determinants sensitive to proteolytic enzyme IgM and IgG. The structure of Ig: joining chain, secretory piece SP, secretory component J chain. Joining chain produced by plasma cells, functions linker to compose dimer, pentamer or polymer. Then secretory piece SP produced by mucosal epithelial cells, secretory IgA, functions protect IgA resistant proteolysis in extra secretory liquid. Domains of Ig. Domain. Polypeptide chains of Ig are folded Polypeptide chains of Ig are folded into a globular structure by intra-chain SS bond within each 110 amino acid region, which is called a domain. Domains of Ig: L chain, Vl, Cl, H chain, Vh, Ch1, Ch2, Ch3, Ch4 in IgM and IgE hinge region. Antigen binding hinge biological activity. Functions of each domain: Vh, Vl, antigen binding site; Ch1, Cl, allogenic marker; Ch2, Ch3, complement fixing site, permeable placenta; IgG, Ch3, Ch4, cell binding site. Hinge region: flexible and suitable for CDR of Ig binding to antigenic determinants. Hydrolytic fragments of Ig. Ig can be digested by papain and pepsin. Position fragments function. Digested by papain. Position near the SS bonds of H interchains of form the end fragments. Fab fragment antigen binding. Fragment crystallizable. Function Fab recognize and bind Ag. Fc fixed complement crossing the placenta bind to FcR in different cells. Antibody exists. Prozone equivalence, optimum proportions of antigen and antibody. Antigen excess. Then digested by pepsin. Position near the SS bond of H interchains from the CN. Fragments and function. FAB find antigen. 2 valence, no function. Significance: elucidating the relationship between the structure and function of IGs, decreasing the immunogenicity of Ig for clinical treatment. Sectional characteristics and functions of the 5 classes of IgS. IgG, highest concentration in 75% of total Ig. 4 subclasses: IgG1, IgG2, IgG3 and IgG4. Unique Ig that can pass through placenta. Half-life is longer. Starts to be produced at 2 to 3 months after birth and reach the level of adult at 5 years old. Functions of IgG: against bacteria and virus, neutralize toxin, combine with the Fc receptor, Fc gamma receptor, activate complement, combine with SPA, some belong to the autoantibodies. Take part in type 1 and type 2 hypersensitivity. IgM, highest pentamer, 10 valencies, J chain, half-life is shorter. The first Ig to be synthesized appear in the early stage after infection, be produced during fetus. The first MIG of the B cell act as antigen receptor, BCR. Functions: IgM is more effective in binding Ag and activating C and play an important role in anti-infection. Then natural AB for blood type antigen. Autoantibody rheumatoid factor, RF. Take part in type 2 and 3 hypersensitivity. IgA, 2 types: serum type monomer, secretory type dimer, trimer or polymer. IgA1, IgA2, secretory is J2, to be produced at 4 months after birth. Exists in almost all body fluids. Which one? IgA. Local mucosal immunity, immune barrier, neutralize virus or toxin, rich in cholesterol, then activate C by alternative pathway. Take part in type 3 hypersensitivity. Then IgD, the concentration in serum is low and sensitive to proteinase. IgD, the concentration in serum is low and sensitive to proteinase, act as antigen receptor on B cell. MIgD, regulate the differentiation of B cells. Then IgE, concentration of IgE in serum is the lowest in normal individual but is very high in some patients, related to hypersensitivity, Fc epsilon R1, mast cell, basophil. Section Fc receptors for AB molecule. Recognize and bind to antigen specifically. Fix complement, bind to Fc receptor on some cells. Recognize and bind to antigen specifically. Fix complement, bind to Fc receptor on some cells. Transfer selectively, placenta transfer, mucosa transfer. Affinity and avidity. High affinity, low affinity, affinity, avidity. IgM-G classical pathway, IgA, G4, GE alternative pathway. Opsonization IgG, IgM enhance the phagocytosis. ADCC antibody dependent cell mediated cytotoxicity, hypersensitivity type cell, mast cell, basophil, FCF, epsilon R1. Section 5 immunogenicity. Isotype CHCl. Isotypic determinants. Mouse IgG1, mouse IgM. IgG1, IgG2, IgG3, IgG4 disulfide bond. Allotype CHCl. Allotypic determinants. Idiotype VHVL. Anti-idiotype antibody. Then artificial lab. Polyclonal AB, monoclonal AB, gene engineering AB. Polyclonal AB, a mixture. Polyclonal AB, a mixture AB with different specificities and affinities generate in a natural response to artificial immunization. Cross reaction. Cross reaction, if two antibodies share an epitope, an antibody recognizes an unrelated but chemically similar epitope. Monoclonal antibody may be produced by single clone or one hybridoma clone and having a single specificity. Prepared by hybridoma technique, immunized spleen cells hybridized with myeloma cells, hybridomas. Artificial antibodies, polyclonal, derived from different B lymphocyte cell lines, batch to batch variation affecting AB reactivity and theta. Not powerful tools for clinical diagnostic tests, but monoclonal, derived from a single B cell clone. MAB after reproducible, predictable, and potentially inexhaustible supply of AB with exquisite specificity. Enable the development of secure immunoassay systems. Monoclonal antibody production, immunization of mouse to stimulate antibody production, antibody forming cells isolated from spleen. Tumor cells are grown in tissue culture. Antibody forming cells are fused with cultivated tumor cells to form hybridomas. Then hybridomas screened for antibody production, hybridomas cloned, monoclonal antibodies isolated for cultivation. Cell fusion technique. Secrete AB short life, no AB long life, myeloma cell, splenocyte B cell, secrete AB long time, hybridoma cell. Screening and hybridoma cell, selenocyte, splenocyte, myeloma cell, hybridoma cell, short life, selenocyte, myeloma cell. HAT selective medium, hypoxanthine, aminopterin, thymidine. Antigen, spleen cells, fusion myeloma cells, culture in hatched medium, test each supernatant for antibodies, clone each positive culture, test each supernatant for antibodies, expand positive clones, propagate in vitro, Applications of monoclonal antibodies, diagnostic applications, biosensors and microarrays. Biosensors are and microarrays. Therapeutic applications, transplant rejection, cardiovascular disease, cancer, infectious disease, inflammatory disease. Clinical applications, purification of drugs, imaging the target. Then future applications against bioterrorism, fight against bioterrorism. Then cell suspension, laser beam flow cytometer. Cell suspension, staining of membrane, cytoplasm and nucleus, laser beam, cell sorting, flow cytometer, cell culture analysis, etc. Evolution of monoclonal antibody. First murine, second chimeric, third humanized, fully human. Gene engineering antibody. Antibody prepared by the method of gene recombination. Chimeric AB, human FC bind with mice Fab, recombinant the single chain AB, VH linker VL. Human mouse chimeric AB,